Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I hope you are all fine. Let's resume the topic where we left it in the previous lecture. We were discussing the life cycle of bacteriophage, which is of two types, lytic cycle and lysogenic cycle. In the lytic cycle, bacteriophage develops a master-slave relationship with its host and brings about the lysis of its host, releasing its own progeny in large number. Whereas in the lysogenic cycle, the case is different. The phage enters into the body of the phage, DNA enters into the body of the host in the same way as in case of lytic cycle, like in this step 1 and step 2, you can see attachment and penetration occurs, but in the step 3, as you can see, this is step 3, integration of phage DNA into the bacterial DNA, because this step is different. Here the phage DNA harmlessly becomes a part of host DNA, so it is called as prophage. Now this prophage, along with the host DNA, it can multiply or it can replicate, and these bacterial cells may exhibit new properties because of certain genes which may be expressed by the phage DNA in the host cell. So this is lysogenic cycle. In the lysogenic cycle, you can see certain terms like prophage. It is the integrated state of viral DNA which enters into an inactive stage, and this is called prophage. Temperate phages are special DNA phages that undergo absorption and penetration but are not replicated or released immediately. The temperate phages are also known as lysogenic phages. Lysogeny is the phenomenon of lysogenic life cycle of bacteriophage. Lysogenic conversion is a term which you will find on page 138 in the site information. You can see in that box when a bacterium acquires a new trait from its temperate phage, because although the prophage is in an inactive state, being incorporated into the host DNA, but sometimes this phage DNA may begin to express its own genes, due to which certain toxins or certain chemicals may be produced in the host bacterium, due to which bacterium acquires new characters or traits. This is called lysogenic conversion. The example given in the site block is of Clostridium botulinum. Clostridium botulinum is a causative agent of food poisoning. Generally, it may be found in the tinned food if it is expired. So it makes several different toxins, two of which are actually encoded by prophage genomes. On page number 139, you find the last paragraph of this replication of bacteriophage in lysogenic cycle. As you can see in these few lines, the lysogenic state of a bacterium can get terminated any time when it is exposed to adverse conditions, and this process is called induction. So induction is basically the activation of the bacterium, the phage DNA, because conditions that favor the termination of lysogenic state, like desiccation, which is dehydration, deficiency of water, exposure to ultraviolet rays or ionizing radiations, exposure to mutagenic chemicals. Mutagenic chemicals are those chemicals which may cause mutations. The separated phage DNA then initiates lytic cycle again, resulting in the lysis of bacterial host and release of new phages or new phage progeny. Such phages are then capable of infecting new susceptible cells and render them or make them lysogenic. So this lysogenic cycle may continue in this manner. Now just have a look at the comparison between lytic and lysogenic infection, or the difference between the two. In the lytic, in the lysogenic cycle, you see it does not kill the host cell outright, whereas in the lytic cycle it kills host cell within minutes of infection. In the second point, viral genetic material integrates with the host genetic material and replicates as host cell reproduces. Viral genetic material is replicated by hijacking host cell in lytic cycle. We have already discussed that. And this third point is lysogenic cycle. In the lysogenic cycle, phage can lie dormant for years, causing no apparent symptoms in the host organisms. Whereas in lytic cycle, virus, that is phage, can cause symptoms in host organism as a result of lysis. Examples: temperate phages, certain cancer causing viruses, they follow lysogenic cycle. Whereas a virulent phage is that which may cause disease or infection that replicates in lytic cycle manner. In the same way, common cold virus replicates in the lytic cycle manner. Now, usage of bacteriophages in genetic engineering. There are different uses of bacteriophages in different fields, but here we are discussing the specific field, that is genetic engineering. First of all, you must know what is genetic engineering. It is the field of biotechnology in which manipulation of genes occur or alteration in the genetic material of an organism is carried out, such as transfer of gene from one organism to another. Several biological tools have been used in genetic engineering, but the bacteriophages are one of those which are most commonly used. Bacteriophages may be used as vector in genetic engineering techniques, just like bacterial plasmids. Plasmids are the extrachromosomal rings of DNA found in bacteria, and you will study about that in the next chapter also. That plasmids are used as vectors in genetic engineering means genes are inserted into the plasmids and then they are carried to the organism where the genes are to be inserted or introduced. In the same way, the viruses or phages are also used as vectors. They are also used to construct or to develop a genomic library. A genomic library is a collection of bacteria or phage clones which contain multiple copies of all the genes of an individual's genome. What is genome? Genome is the complete set of genes of an organism. So all the genes of an individual may be attached to the phages or may be attached to the plasmids, and those are then inserted into the bacteria where they may be kept in the gene banks, and they can be utilized in different research purposes or in the treatment of or diagnosis of different diseases also. Next point is phage therapy. Phage therapy is another important usage of bacteriophages. It is the application of genetically engineered phages that can kill pathogenic bacteria. Phage therapy has advantages over conventional antibiotic therapy. Antibiotics are least selective. So they do not destroy selective bacteria, but they may damage your good bacteria also, which constitute your normal bacterial flora. They are found in your intestines or different parts of the gut, and these bacterial flora which are useful bacteria or good bacteria in our body. They produce different vitamins, etc., in our body. So the antibiotics, they may also kill the useful bacteria along with the bacterial pathogens, whereas phage therapy is useful in the sense that it is highly selective and it will destroy only the specific bacteria, as we have discussed in the earlier topic that phages or the viruses are highly host specific. Next point is the development of antibacterial nanomedicines. What is a nanomedicine? First of all, you must know what is nanoscience. It is the study of those structures that are between 1 and 100 nanometer in size. So nanoparticles may be found in different materials like the ash in the like ash or fine sand, etc., and the nanoparticles are also found in the viruses. So the viruses or the phages are used as a tool for the treatment of bacterial infections, but the use of phages as nanoparticles, they are used to be formulated as targeted drug delivery vehicles. What is the meaning of this statement? It means that the phages help in the delivery of drug to the specific target site, which may be specific target cells, as phages, they are highly specific in their host, so phages may be used to deliver the drug effectively to only selective cells. So this is very important development in the nanoscience. With this, we come to the end of this chapter. Just for your interest, I have added a song, which is a scientific song in the lecture. So enjoy it, and Allah Hafiz till the next lecture.
I'm a virus, an infectious particle, an anathugger pirate, the genuine article, Ebola, chickenpox, West Nile, influenza, yellow fever, AIDS, herpes, are coming right at you. I'm not a cell, not an independent organism, don't even have my own metabolism. I only reproduce myself by taking over cells, then I bust them apart, the one that you reveal well. The structure, the simple, mostly proteins and genes, packaged as a sub-micron killing machine. My capsule's made of capsules and self-assemble. Whenever cells see me, they start to tremble inside my capsid is where my genes reside. I got DNA or RNA on the inside, and the rest of me is various proteins. Role is assisting in assault cells and taking control of the virus.